Okay. Let me know if everybody can see that. Let's see. Get it started here. So I've got some in a dish here. Get it in focus. So you can kind of see their little eyes. There's a part, the pharynx, which they use to eat. Let's see if I can point it out to you. Is right in this area. Let's see. A whole bunch of them here. I don't know that I have any with a blastema right now, but the way these guys reproduce is actually by fissioning. So what they do is they have kind of an adhesive gland on their tail, and they'll stick that down to the substrate, whatever they're on, in this case, a plastic dish, and attach it, and then they'll actually tear themselves in half. So they'll tear off little pieces of their tail, and like in the slide I showed before, each of those little pieces will become a new worm. So they're not going to do that for you right now, probably, but I can kind of, we can accelerate this process. The way we sort of work with them in the lab, if we want a whole bunch of them, we'll just cut them up. So I can kind of show you what I'm talking about. It's too bad you guys can't come in to do this yourselves, but. So there's a tail fragment. And if you watch like the fragments, the headpiece will keep, swimming around. Let's see if I can zoom in for you. And then the body piece is sort of swimming away. So they kind of have they don't necessarily need the brain to exhibit gliding motion. They need it for certain behaviors like light avoidance. But in a lot of ways, the pieces will behave autonomously on their own, like even after you cut it. So we can cut up another few more. Let's see. Come back.
Tom, there's a question.
Oh, yeah.
Sorry, I can read it for you. It says, what happens if you chop them vertically rather than horizontally?
Ooh, yeah. Well, let's see. Let's try it. It's sort of a bigger wound face. I'll show you what happens immediately. I can tell you what happens in the long run. Each of the pieces you cut will become, they'll also become a new animal. It's just instead of regenerating, let's see. Okay, that was kind of a bad job, but sort of get the idea. So they'll regenerate laterally is what they'll do. The left piece with a cut on its right will regenerate a right side, and the right piece with the cut, left-facing cut will regenerate its left side. It does maybe put a little bit more stress on the animals to be cut that way because there's such a large wound face, but they will regenerate most of the time. Something you may notice, I don't know if any of you can kind of see they have pigment cells, and as you, oops, I lost them. You may notice that a few minutes after getting cut, you can see sort of a dark band start to appear like near the cut face. I don't know if you can see that in this one. Sometimes it takes a few minutes. And what's happening there is all the muscle cells. So these animals have different types of muscle. A lot of the muscle that they use for sort of motion is right under the skin, and that muscle will contract. It'll start contracting to try and close up that wound face, so it's not exposed to the water. So that's kind of what happens if you make that sort of cut. It'll regenerate though. All right. Does anybody want to see? Let's see if we can. I have something else that might be kind of fun, just to show you their anatomy. So we normally feed them liver, like crushed up beef liver. And so let me see if I can get them to eat, because the way they eat is kind of cool. Just one sec. All right, so I just put a little bit of, this is a little bit of liver. Let's see if they get interested in it. Hopefully you can see in a second the way they eat using this sort of interesting pharynx structure. This guy might be interested. Oh yeah, okay. Can you see the pharynx there? He's sort of on top of it right now. Oh, there it is. Let's see if I can. So he's sort of vacuuming up all that the beef liver. It's a little bit hard to see because the blob of liver is in the way. You can, right there, you can see the pharynx coming out of that one. It's kind of hunting around for it. The interesting thing about the pharynx is it'll regenerate too if you cut it off. And it'll keep moving sort of autonomously on its own for a while, and it'll actually continue making like eating motions, even if you cut it like off of the animal. So it has sort of almost like a nervous system of its own, like a mind of its own, kind of almost.
Tom, we have another question.
Yeah.
Which is, do they ever die?
Very good question. That's a great question. We have a student in the lab right now who is actually working on a PhD related to that topic. So this is an active research area that we don't like quite fully understand yet, but theoretically no. They have a stem cell population that is capable of producing any cell type in the body, and those stem cells self-renew pretty much indefinitely. Unless the animal gets sick due to some bacteria infecting it or something like that, those stem cells self-renew sort of perpetually. One interesting thing about them is, I don't know if any of you have heard of telomeres. It's a type of DNA that kind of caps the end of the chromosome. Most animals have linear chromosomes. Bacteria have chromosomes that are a circle generally. Some don't. But most, animals have linear chromosomes. And at the very end, there's this structure that's really important called the telomere. And as humans age, studies have shown that the telomere gets damaged. It gets shorter and shorter. And if it gets too short, you run into problems of proper segregation of the chromosomes, proper maintenance of the chromosomes, and those cells eventually senesce, or in other words, the cells become like old. But these guys, Schmittea, these planarians, maintain long telomeres throughout their life. So every time the neoblast, these stem cells in them renew, they renew the length of the telomere. So as far as we know, they're immortal. So it's kind of difficult. It's difficult in some ways to study something like that, but yeah, great question. As far as we know, they're immortal. Seems like a lot of them are getting really interested in feeding now. Okay, let's see. Okay, so I wanted to show you guys a couple more slides to kind of describe some of the types of questions we are interested in. We'll go back to the microscope in a minute here, but let me share some other slides with you. Just one sec. Okay. Right. So this is kind of a time course showing how regeneration happens. Now, something that was noticed, planarians have been studied for, at least 100 years. One of the things that was noticed pretty early on is if you make these fragments, this is a pretty big fragment. If you cut smaller and smaller pieces, sort of smaller and smaller slices of the animal, what was noticed was that sometimes instead of regenerating a proper head and tail, if you made the piece really small, you would get animals that have, 2 heads or 2 tails. And the sort of genetic tools of the day, it was difficult to really figure out why that might be. But people had a theory, sort of put forward this theory that it might be due to what's known as a morphogen. So I'll sort of touch briefly on what that is. Morphogen is like something a cell releases, like a protein or some type of small molecule a cell releases to communicate with distant cells. So in other words, if we imagine one cell here releasing some type of compound, where if we look at like distance versus concentration of the compound, it kind of tails off because it's diffusing, right? It's like some type of protein that's diffusing away from this source cell. And scientists imagine that maybe what is happening to control the head versus tail of the animal is some type of morphogen gradient. In other words, they imagine cells in the head might be releasing some type of compound that diffuses away and then is at a lower concentration in the tail, or maybe the other way around, where something is released from the tail, controlling tail identity, that gets less and less concentrated as you get near to the head. So this was kind of an interesting, alluring idea, but no one knew if this was true or not until more recently, where this is some data from a study from our lab from a number of years ago. There are these genes that are secreted from the cell. Some proteins spend their life inside the cell, but some are released from the cell. So this gene called Wnt, or family of genes really, is like that. And this postdoc in the lab, Chris Peterson, found that there are certain Wnt genes that show this gradient-like pattern, where it's at high concentration in the tail and then low concentration in the head. And so this was very interesting because it sort of gets at that idea of, original idea of like, if you cut a very small piece, why might that piece get confused sometimes? And the thinking is, well, maybe you can't really see the gradient if the piece is too small. There's not going to be enough of a difference between the front of the piece and the back of the piece. Whereas, because you need like a bigger piece to see the full sort of difference. And so what Chris did and others was sort of test whether these Wnt genes, this Wnt pathway, was involved in controlling head versus tail. And this is a really cool result. He found if you knocked out the activity, if you reduce the activity of a gene that's needed for Wnt signaling called beta-catenin, you get these animals that when you cut them, they regenerate 2 heads instead of the proper head and tail. And it just so happens that someone in the lab, they're not mine, but has a couple of these animals for some experiment that they're doing. So I can show you, it's kind of neat. Let me switch to my video again. Hold on. Let's see. Find some. Here's one. This one, the eyes on the one side are kind of funny, but you can sort of see that it has eyes on both sides. Let's see if I can find another animal. This may be a better example. Here's a good example. See, can you kind of see? Oh, hold on just a sec. Where'd you go? Squirt it so it shows. One sec. They like to hang out around the edges of the dish, so sometimes you have to like coax them into the middle. Let's see. Yeah, this is a good example. Yeah. So you can kind of see eyes on both sides there. Sometimes they, with these multi-headed animals, you can see that you can kind of tell from the way it behaves that it has two brains, and they're kind of like competing with each other for the control of the animal. In this case, it seems like and one of them gets kind of dragged along. It's sort of interesting. So this is the beta-catenin animal. And like I was saying, what that means is we use this technique called RNA interference. Basically what you do is when you want to disrupt the activity of a particular gene, you create a double-stranded RNA. You know, in your biology classes, you may have heard of the central dogma, which means essentially gene activity starts with DNA, and then DNA is transcribed into RNA, which is then translated into protein. So what you can do, it was found, if you make a double-stranded RNA and feed it to these animals, the RNA gets taken up by the cell and sort of chopped up and then used by a series of enzymes. Dicer is one of them. They take those RNAs that you feed them and they use them to target RNAs inside the cell for destruction. So in this case, when we feed them beta-catenin RNAi, or double-stranded RNA, it targets beta-catenin in the cells of the planarian for destruction. So it sort of disrupts the activity of that gene.
Tom, quick question. Can they ever pull apart from each other?
Oh, those two-headed guys?
Yeah.
You know, I haven't worked myself with them too much. Maybe. The way they pull apart with just wild type animals is they stick the tail down and so they sort of like, I don't know if they would have enough like to get enough sort of leverage to pull apart if it was just like sort of two heads pulling. They might need like a tail in the mix to get that to happen. Not exactly sure.
Yeah, there was another question actually. It's interesting because someone was asking, would the worm die if you smushed it? So that being, I mean, it's a thought, right?
You know, that's kind of a, I'm glad you asked that. People in the planarian field had often wondered whether when they fission, in other words, when they rip themselves apart, whether it just happened like at random, they broke in random places, or whether they broke in like specific sort of prearranged planes. And this lab did kind of a wacky experiment, sort of a fun experiment. And I think they did it by accident, by smushing an animal by accident. They found that when you smush them, a lot of the times, like sort of very regular planes sort of fission planes would appear, and it would kind of break into, roughly equal sized pieces. It wouldn't just sort of break randomly when you smushed it. And so it's kind of an active area of research then became very interested in like, oh, why is that? Why are these like fission planes appear so regularly? What's going on in those cells? I mean, if you smush it really hard, yeah, it'll probably die. The thing is, like when we cut them they do need some integrity of the skin. Like the cells in the middle of the animal don't like being exposed to water. They are sort of protected by the skin layer and the mucus around it. So if you smushed it, they might be sort of too exposed. They might live. It's hard to say.
Thank you.
Yeah. Okay, so I have some other cool stuff to show you guys. Let's see, let me share that screen again. All right, so that's the kind of beta-catenin story and a little introduction to anterior, posterior gradients, in other words, head to tail gradients and how they control the body plan. And there's an interesting story that is related to that and kind of also related to biodiversity, which I wanted to share with you guys. So Schmittea mediterranea, as you might guess from the name, is from the Mediterranean. But there's also there are a lot of local planarian species that you can find here in Massachusetts. One of them is called Procatula fluviatilis, and you can find it lives in a lot of streams and ponds around here. This is me out on a hike with my dogs, and I figured it would be sometimes I bring like collecting tubes with me. So we were by this little pond, and I stopped by the bank, and you can normally find these guys like under in really shallow water, often under like rocks and sticks and stuff. If you just sort of pick up a rock or a leaf or something that's by the edge of the pond there and turn it over, oftentimes you'll find these guys. Whoops, can I go back? You'll find these guys sort of hanging out under there, and they eat all sorts of little invertebrates that are living in the water. And the reason, one of the reasons anyway, this is sort of a cool species to study, is that unlike Schmittea, the main species that I just showed you, P. fluviatilis can't regenerate a head from the tail fragments. So what I mean is, whoops, no, go back. What I mean is if you cut it here, this is the head, the pharynx, and the tail. If you cut it close to the head, this tail piece will regenerate a head after a few days, which is what you see here. You can sort of start to see the eyes come in. But if you cut it on the other side of the pharynx, suppose you cut it here on the tail, this tail piece will not regenerate a head ever, pretty much. And so it's sort of an interesting species to compare with Schmittea, which can do regeneration from any piece, to ask the question, well, why is that? Why can Schmittea, be so good at regenerating, and this one seems to have some difficulties? So, what was found in this? I keep going back and forth. What was found in this study is they did some RNA sequencing. RNA sequencing is a nice technique to sort of read out the activity, how active a gene is in a given part of the body, by looking at how highly it's transcribed. So is the DNA producing a lot of RNA? Is it transcribing a lot of RNA or a little bit? You can sort of call that gene activity. So what they did is looked at gene activity in these tail pieces right after regeneration versus gene activity in these like head amputated pieces. And what they found was that wints, the same wints that I was talking about before, are much more active in this part here than you would expect from than this part. So there, what you might expect is like, oh, it might. The authors were thinking maybe the reason they can't regenerate a head is because the wind activity is too high. So in other words, like if we, imagine a wind gradient like this, high in the tail and low in the head, maybe if it weren't like this and it were high everywhere, then it would have a tough time regenerating. So as you might imagine, the next experiment they were sort of interested in was, well, maybe if the problem is too much wind, we can feed this animal beta-catenin RNAi to reduce the wind, like was shown in that other paper. So they did that, and it's kind of cool. If you feed them beta-catenin, like the two-headed animals I showed before in Schmittea, you see that all of a sudden, those tail pieces that before couldn't regenerate a head can now regenerate a head. So it's kind of a interesting species, this P. fluviatilis. And I'll show you some in a minute here, although I don't have any mutants. It's kind of an interesting species to compare to other regenerating species to see, like, kind of what makes them tick. Why are they different? Why do they regenerate in different ways? And it's a really kind of nice example of why biodiversity is like important, because it enables us to have all these different species to compare to. So let me show you just real quick some. I think I have some P. fluviatilis here. Let's see. I don't have as many of these guys. I have some. This one's not very active. These ones are a little bit slower than Schmittea. They don't, they're a bit lazier. Let's see if I can find one. Just one sec. Oh, here they are. You can kind of see it. Zoom in some more. These guys, when they hang out near the edge, are a little bit tough to dislodge. They have like, they're really sticky. So once they stick down to something, it's like, they're really stubborn. They have a lot of mucus, and once they stick to something, they really don't want to move. So it's a little, you need like a paintbrush or something, which I don't have at the moment. But in any case, these are, I think I actually collected these guys on that day that picture was taken. So we've got a couple of these in the lab that we're using for various experiments. We've got some other cool experiments with these guys too.
Tom, do you think that's an evolutionary mechanism, so to speak, that one will regenerate a head and one won't? That there's some sort of divergence or something?
I think there's probably an interesting. I don't think it's known. There's been some speculation over like why that might be. I don't think it's known yet, so it's sort of an interesting area, active research area to think about. One thing that's worth noting is they have very different reproductive modes. So the Schmittea that I was talking about, they reproduce mostly by fissioning, just ripping themselves apart, so it's asexual. But P. fluviatilis and a lot of other planarians are sexual. They're hermaphrodites, and so I think actually they only live about a year, and they reproduce once, during the year, lay eggs, and then they kind of die. So it's, it's one of the things that is maybe a reason is regeneration is so important. Being able to regenerate any type of part is so important for Schmittea because it sort of used that as its reproductive strategy. It couldn't have this fissioning mode of reproduction if it wasn't so good at regenerating. Whereas for P. fluviatilis, it mostly just reproduces through laying eggs. So that's one potential reason, although I don't think that's been like definitively proved that that's the the reason for the difference. But yeah, that's a good question. All right. I want to, let's see, I want to kind of close with a series of experiments that I'm doing for my work, which is on another type of regenerating species in the lab. That's kind of cool. So I'll show you some slides on that, and I'll show you some animals, and also do kind of a neat experiment. So just one second. Okay. All right, so here's kind of a phylogenetic tree showing all the different animals. Here's humans, here's flies, here's planarians, and then like sponges and stuff, or hydra. Now, planarians regenerate really well. An interesting question to ask is, like, sort of look across the animals and ask what other animals regenerate really well. And it turns out that there's this group of worms called aceals that can also do whole body regeneration. And it was thought for a long time that aceals actually belong to the planarian group, and they were just a type of planarians. But in recent years, actually quite recently, it became possible to really delve deeper into that question of, are they really a planarian or not, by using DNA sequencing. So it's a little tough to, guess what clade an animal belongs to just based on its physical characteristics, but it turns out to be much easier to tell when you DNA sequence its whole genome and compare the two DNA sequences. So when the sequences were compared, it actually turned out that aceals are not only not a planarian, they're hardly related to them at all. They turn out to have diverged from all the other animals very anciently, like about maybe 500 million years ago. So they're what's known as like a basal clade. They diverge right near the base of the tree. But since they also do whole body regeneration, it's kind of interesting to ask, are the mechanisms the same? Or do they also have neoblast stem cells, and do those stem cells behave the same way, etc.? So the type of aceal that we study a lot in the lab is called Hofstenium miamia. And let's see, I can show you, let me stop the share here to show you a few of these guys. Got some here we can look at.
It's nice to hear, Tom, that the lab is active, that there's people
Yeah.
are working. That's what it's like. So they're getting sort of the experience, maybe virtually, but that, it's, the labs are busy. That's good.
Yeah, we're active. We've been operating at like half capacity, everyone wearing masks, but we're staying active.
Good.
Okay, so these are, whoops, where'd you go? These are some Hofstenia. Let's see if I can get this in focus. And you notice their body plan is a little bit different. They're not actually flat, like the flatworms. They're more like a tube, really. And they also don't have any eyes, you might notice. Although, if you look closely, I don't know if you can see, it does have a single spot in the sort of near the forehead, I'll call it. Sort of like a little dot there. Where'd you guys all go? It's a little bit tough to see under this magnification, but that little dot near the forehead is called a statocyst. This one has maybe a better image. And what it is, it's a gravity sensing organ. Let me stop this. and share. I can sort of give you a better idea through some slides. It's a gravity sensing organ. So what it is, it's like a little bag, essentially, a little sphere. Here's a picture of one in Hofstenia from a former postdoc in our lab's paper. And it has like a little mineralized sort of stone, if you will, like a little rock inside it. And, you know, not a ton is known about how exactly it works, but you can get the general idea. This sort of stone is suspended, freely suspended in fluid within this sort of larger sphere. And you can imagine as it bounces around or sinks to the bottom, it can function as sort of a accelerometer, or, you know, a way of figuring out which way is up, of which way gravity is pointing. And these types of gravity sensing organs are kind of interesting. They're found, some type of gravity sensing organ anyway, is found in almost all types of animals. Now, I should mention that they're not all morphologically the same. In other words, they don't all look exactly the same. Like the human inner ear or the fish, you know, inner ear does a sort of similar function. There's kind of a bone or a mineral inside a larger structure that, you know, transduces acceleration. It's my guess at this point, we're still sort of working on this, is that a lot of these different types of statocysts and gravity sensing organs probably evolved independently, perhaps through the same mechanism, similar mechanisms, or perhaps through completely different mechanisms. But in any case, it's sort of interesting. All animals seem to have them. And this is kind of a close-up of the acial statocyst. You get this mineralized, you know, statocyst, and then like a cell sitting on top of it, and then a bunch of liquid that it sort of floats in, and then another couple of cell types here. So one of the things I'm working on is how statocysts regenerate. And it's sort of interesting to me for a couple reasons. One of them is sort of basic to all regeneration biology, and it's the question of, do the stem cells and, you know, precursor cells that are needed to build any structure really, do they respond to the presence or absence of that structure? You know, if you cut it out, let's say if you cut out the statocyst, do they make more statocyst precursors? Do they sort of like ramp that up to make more, or do they just kind of let it passively regenerate? So that's sort of one interesting question. It's also just kind of fun. You know, gravity is something really universal, and it's just sort of fun to think about how animals sense it and how the genetics of that works. So these are some of the types of things that are motivated my work on this. So some of the types of experiments I'm doing are using RNA sequencing to figure out what genes are important for building statocysts. You can do this by, you know, dissecting them out, cutting out the statocyst versus like some surrounding tissue that we use as a control, and then sequence both and see what types of genes do we see strongly expressed here but not here. And that's something that is maybe a good candidate for being an important gene for making that. And then the second step is to sort of do RNA interference to disrupt the function of those genes. You can do that by sort of soaking these animals in double-strand RNA. And then we amputate them, wait a few days, and then see if they're able to regenerate the statocyst. So these are some of the types of things I'm working on. And I thought in the remaining time here, it might be fun to show you guys a technique. It's kind of a manipulation technique that we can use to study the regeneration of these statocysts. We have in the lab a laser ablation. It's called a laser ablation system. It's like a pretty strong laser that you can, you know, zap, but it's very focused. You can sort of kill a specific cell type. So I can take some of these just walk you guys over there just one sec, and we can test it out. Let's see.
Thomas,
Sorry, I hope you can hear. There's like a loud freezer.
oh, we can hear. It's
Oh, yeah,
nice to see the Whitehead again, though. I haven't. I'm not there that often with COVID. I've been working from home, so it's nice to see the space.
All right, so here's kind of the setup. It's just like a regular microscope, except it has like a laser connected to it that sort of feeds laser light in through the microscope objective and focuses it on the sample. Let me make a little slide. Essentially what I'm going to do, you can see I've got some animals here. I'm just going to put one of them on the slide. Then you'll get to see a better picture of the status system. We can also try to ablate it. Just one sec. Right. All right, so I've got like a little slide. Oh, it's a little hard to see, but I've got a slide here with an animal on it. Fire this thing up. Let me start my screen share so you can see. Kind of smushed itself. All right, I think it's a little bit upside down, but you can sort of see, zoom in. Kind of see the status there. It's a little hard to see it because it's upside down. But let's see if we can get a shot in here. Turn on the laser. Oops. Okay. I'm having a little bit of tough time hitting it squarely, but that big bubble there you see is sort of some damaged cells from one of the laser pulses. Oh yeah, I got a good shot in there. All right, so now what we can do I'll stop my share, pull it out, rescue it kind of into some fresh seawater. Just 1 sec. And then take it back. Shut this off. Sec. Oops, sorry, Jen. All right, so I don't think I have enough time to show you guys, but the actual assay, but I'm also studying the behavior of these guys. So after doing a laser ablation like that, we'll take it, put it back into like some fresh seawater so that it's sort of happy again. And then I have a behavior assay, which I'll show you here. It's sort of like a glass tube at an angle. And what I do is put the animal in there, and they tend to exhibit, the wild type animals, the unaffected ones, will exhibit like a downward swimming behavior. It's a little bit unclear exactly why they do this, but my sort of rationale is if these animals are in the wild and they become, exposed on the sand or something like that, they would want to swim downwards to sort of get into deeper water where they're more safe. In any case, it's a pretty reliable, like, sort of gravity-focused behavior that we can assay for, after doing staticist ablation with a laser or with after various gene knockdown by RNAi. So that's some of the work we're doing to kind of focus on, neural regeneration, staticist regeneration, and how that happens, the genetics of it. Okay, so I think that's about all I have for you guys. If you had any questions, I'd love to hear from you and field questions if you have any.
Tom, I had one come up when you were at the laser.
Yeah.
Someone was asking, is that used for heart procedures, or at least a similar setup?
I know lasers are used a lot these days in like microsurgery. I mean, certainly I know a little bit about like lasers in eye surgery. My wife got like laser eye surgery. Heart, maybe. It's certainly, I think it's used in a lot of areas of surgery because it's so precise. I mean, not that specific setup. This microscope setup is sort of more specific to basic research. I think a lot of the lasers used in medicine are like little handheld or like robotic arm held lasers. But yeah, I think they're used a lot.
Thank you.
Yep.
Does anybody have any questions for Tom? Or again, if you have questions about sort of his specific career path or how he got interested in what he's doing, feel free to ask that as well.